Genetic transcription and translation is pretty involved, and it requires several players: DNA, messenger RNA, ribosomes, and transfer RNA. DNA, of course, stands for deoxyribonucleic acid and is found as a double helix in the nucleus of the cell. DNA contains the genetic material that runs all processes in the cell. Protein synthesis begins with a process called transcription. The DNA code is transferred to messenger RNA in the nucleus of the cell. RNA polymerase creates a messenger RNA strand using the DNA as a template. This new messenger RNA moves through the nucleus to the cytoplasm. Messenger RNA associates with the ribosome subunits, and an enzyme in the cytoplasm activates its transfer RNA by attaching a specific amino acid to each transfer RNA. The first transfer RNA attaches to the A site of the ribosome and begins a sequence of building a protein using amino acids. In this scenario, the first transfer RNA carries the anticodon UAC, which is complementary to AUG on the messenger RNA strand. Then, the ribosome moves down the
Chargaff's rule.
Transfer RNA messenger RNA complex. The first transfer RNA anticodon is now in the P site on the ribosome. A new transfer RNA, complementary to the next anticodon in the sequence, moves into the A site, carrying the next amino acid. In this example, the transfer RNA has an anticodon AAA, complementary to UUU on the messenger RNA strand. An enzyme in the ribosome catalyzes the formation of a bond between the two amino acids now in the ribosome. Then, the ribosome moves down the transfer RNA messenger RNA complex again. The transfer RNA that was in the P site is now in the E site, and the transfer RNA that was in the A site is now in the P site. The transfer RNA in the E site doesn't have an amino acid attached anymore, so it is released. This process also opens the A site to accept the next transfer RNA. This shifting and adding continues until the ribosome reaches the stop codon, signaling the end of the protein synthesis.
Section 3's main idea for Chapter 12 is DNA codes for RNA, which guides protein synthesis, as you saw in the video. There's quite a few vocabulary words that you will need to not only know, but the definition, and also be able to use those. The central dogma. After the discovery of DNA structure, scientists turned to investigating how DNA served as a genetic code for the synthesis of proteins. Geneticists accept that the basic mechanism for reading and expressing genes is from DNA to RNA to protein. This is referred to as the central dogma of biology, meaning DNA codes for RNA, which guides the synthesis of proteins. This bullet point right here, this section is very important, guaranteed to be on your EOC. RNA is a nucleic acid similar to DNA, but with the sugar ribose and uracil instead of thymine. Messenger RNA, otherwise known as mRNA, are long strands of RNA that are formed complementary to one strand of DNA, direct synthesis of a specific protein. rRNA, which is ribosomal, associates with proteins to form ribosomes in the cytoplasm. And tRNA for transfer, smaller segments of RNA that transport amino acids to the ribosome. So let's talk about transcription. It's the first step of the central dogma, and it involves the synthesis of messenger RNA from DNA. The enzyme RNA polymerase regulates RNA synthesis by binding to the specific section where an mRNA will be synthesized. RNA polymerase moves along the DNA strand in a 3' to 5' direction, synthesizing mRNA or messenger RNA. Once that happens, RNA processing, in comparing DNA code with its transcribed mRNA code, scientists found that mRNA code was much shorter. DNA sequences not found in messenger RNA are called introns, and DNA sequences that remain in the final messenger RNA are called exons. The code. Scientists hypothesized that the instructions from protein synthesis were encoded in DNA. Their 3 base code in DNA, or mRNA, is called a codon. So, UUU is a codon. UCC is a codon. UCG, that's a codon. After synthesis, messenger RNA moves from the nucleus into the cytoplasm where it connects at the 5' end to a ribosome. The messenger RNA code is read and translated into a protein through a process called translation, and this is what you saw in that video. tRNA molecules act as the interpreters of the mRNA codon sequence. The tRNA is activated by an enzyme that attaches a specific amino acid to the 3' end. The middle of the folded tRNA contains an anticodon, a complementary sequence to the mRNA. Ribosomes provide a site for protein synthesis, which is our endgame. When mRNA leaves the nucleus, the two ribosomal subunits come together to hold the mRNA in place for translation. The ribosome structure has grooves that hold and serve as tRNA sites for amino acid attachment.
When we start learning about protein synthesis, we usually use part of the DNA molecule to act as a pattern for transcription and translation. As the beginning of transcription. And so this is half of my DNA molecule, and I would like to know what mRNA is going to bond next to this DNA in the nucleus during transcription. So I would have G for guanine, C, A, G, G, U, A, and G. So this is my messenger RNA. Messenger RNA comes into the nucleus and copies the DNA code. So that's my RNA will now take this code out of the nucleus and it will arrive at the ribosome. So now let's say that messenger RNA has arrived at the ribosome and transfer RNA is going to bond next to the messenger RNA. So that will be C, G, U, C, C, A, U, U, C. And that's my that's my transfer RNA, excuse me. So this green, this is my transfer RNA. And I might ask, how many amino acids is this genetic code going to code for? Basically, every 3 bases in a row is going to be carrying 1 amino acid. So this would code for a particular amino acid. This one would code for a particular amino acid. And then this one would also.
After learning about DNA, have you ever wondered how can the DNA actually result in a trait? Let's take an example, like eye color. Yes, your DNA has the genetic information that codes for the color of your eyes. Your eye color is based on a pigment that is inside the eyes, but in order to have that pigment, you have genes, which are portions of DNA that can code for proteins, which help make that pigment. So what we're going to talk about is how your DNA can lead to the making of a protein. This process is called protein synthesis. Synthesis essentially means to make something. So protein synthesis means to make protein. And you may wonder, what's the big deal about proteins? Well, you may not realize this, but proteins are kind of a big deal. They do all kinds of things. Proteins are involved in transport, in structure, in acting as enzymes that make all kinds of materials, in protecting the body, and so much more. You've got to make proteins. It's essential for you to live. And what is so cool is that you are making proteins right now as you sit and watch this video. It's happening in your cells. They're making proteins. So back to your DNA and its role in all of this. All of your cells have DNA. Well, a few.
There's a video titled Why RNA is Just as Cool as DNA, and I will be uploading that as a second portion to 12.3.
Protein synthesis, we can look at 2 major steps. One is transcription and the other is translation.
Transcription has a C in it, and translation has an L in it. I remember that C comes before L in the alphabet, which helps me remember that transcription comes first. I like a lot of alphabet mnemonics. Now, transcription is when we're going to transcribe the DNA into a message. In your cells, the DNA is in the nucleus, so therefore we're doing transcription in the nucleus. In the step of transcription, an enzyme called RNA polymerase will connect complementary RNA bases to the DNA. These RNA bases are bonded together to form a single-stranded mRNA. The M in mRNA stands for messenger. Messenger RNA consists of a message made of RNA that has been based on the DNA. We do want to mention that this mRNA is not usually ready to go right away. There's usually a significant amount of mRNA editing that occurs. We highly encourage you to do some reading about that because it's not only fascinating, it's critical for the process to work correctly. So what's something great about being mRNA? Well, in eukaryotes, you get out of the nucleus. The mRNA can go out of the nucleus into the cytoplasm where it's going to attach to a ribosome. Ribosomes make protein. The ribosome is made of rRNA, and that's an easy one to remember because the R stands for ribosomal RNA. The ribosome is going to build our protein in the next step called translation. You know, you can find a lot of great clips and animations on translation that are just fantastic. We're just going to break down some basics of what's happening. In the cytoplasm, if you look at this, you have all these tRNA molecules available. tRNA stands for transfer RNA. They carry an amino acid on them. An amino acid is the monomer for a protein. If you have a bunch of amino acids together, you can build a protein. So it's the tRNA that is going to bring those amino acids together to make that. But wait, how does the tRNA know which amino acids to bring? That's why the mRNA, the message, is so important because it's going to direct which tRNAs come in and therefore which amino acids are transferred. It reads the bases, represented by these letters here on the mRNA, in threes. So it doesn't read one letter at a time, it reads in triplets. That's called a codon. These amino acids are held together by a peptide bond, and it will keep on growing. Typically, at the very end of the mRNA, there is a stop codon. Stop codons do not code for an amino acid, but when the ribosome reaches it, it indicates that the protein building is finished. So the result of translation is that you built a chain of amino acids that were brought in certain sequences based on the coding of the mRNA. But remember, that mRNA was complementary to the DNA, so the DNA ultimately was the director of the entire protein building. Of course, it couldn't have done it without some serious help from mRNA, rRNA, and tRNA. Protein folding and modification may occur, and the protein may need to be transported. This can all vary based on the protein structure and function.
So the Beetle and Tatum experiment showed that one gene codes for one enzyme, and we now know that one gene codes for one polypeptide.